Bismillah ar-Rahman ar-Rahim. I will continue about the cellular reaction to an injury. Lecture 2 includes pathogenesis of ischemic cell injury and irreversible cell injury, necrosis, and types. What we said pathogenesis, we mean by the mechanism of the development of a disease. The cellular response to an injury depends on the type of the injury and severity. Low doses or toxins of the toxin or brief ischemia may lead to reversible cell injury, whereas larger ischemia may result in irreversible cell injury and cell death. The same injury has different outcomes. Striated muscle in the leg accommodates complete ischemia for 2 to 3 hours without death, whereas cardiac muscles dies after only 20 to 30 minutes. The principal targets and biochemical mechanism of cell injury are mitochondria and their ability to generate the adenine triphosphate ATP and reactive oxygen species, disturbances of calcium homeostasis, damage to cellular plasma membrane and lysosomes, and lastly, damage to DNA and misfolding of protein. This figure illustrates the principal biochemical mechanisms and sites of damage in cell injury. Mitochondrial damage will decrease the ATP, increase reactive oxygen species, and entry of calcium will lead into an increased mitochondrial permeability with activation of multiple cellular enzymes. Cellular or membrane damage will lead into a plasma membrane loss with damage with loss of cellular components. Lysosomal damage will lead or rupture will lead into enzymatic digestion of cellular components. We call it autolysis. Protein misfolding and damage will lead into activation of pro-apoptotic proteins, and I will speak about this in the next lecture. First, we will speak about the effect of damage to the mitochondria on the ATP. Whenever there is ischemia or decreased blood supply to the tissue, there will be a mitochondrial damage, and this will lead into a decrease in oxidative phosphorylation. This will lead into a decrease in the ATP. Decreased ATP will lead to a failure of many energy-dependent cellular systems, including the sodium pumps is reduced, resulting in intracellular accumulation of sodium and efflux of potassium, accompanied by osmotic gain of water, causing cellular swelling. So there will be first effect is on the sodium pump, will lead into decreased sodium with efflux of potassium outside of the cell with influx of sodium, water, and potassium inside the cell. This will lead into a cellular swelling, followed by endoplasmic reticulum swelling with loss of microvilli and blebs. What do we mean by a loss of microvilli with blebs formation? As we know, the plasma membrane has this microvilli. Whenever there is influx of water and sodium, the cell got swollen and there will be a formation of a blebs فقاعات. There is a compensatory increase in the anaerobic glycolysis in an attempt to maintain the cell's energy sources. As a consequence, intracellular glycogen stores are rapidly depleted. So decrease ATP will stimulate and increase anaerobic glycolysis with decrease of glycogen. As we know, decreased ATP, as we said, will stimulate anaerobic glycolysis. How? AMP, adenine monophosphate, gives adenine diphosphate and later adenine triphosphate. Whenever there is a decrease in ATP, there is an accumulation or increase in AMP. This will stimulate the phosphofructokinase, stimulate anaerobic glycolysis with decrease of glycogen. Also, decreased ATP will lead into increased lactic acid. This will lead into a decreased pH of the cell with clumping of nuclear chromatin. Other effects of decreased ATP, detachment of ribosomes with decrease in protein synthesis. Other effects of a mitochondrial damage, as we said, it will lead into an increase of reactive oxygen species, and this will lead into a lipid peroxidation with membrane damage, protein modifications with breakdown and misfolding, with DNA damage and mutations. The role of increased cytosolic calcium in the cell injury is demonstrated in this figure. Whenever there is increase in cytosolic calcium, there will be an activation of cellular enzymes. Phospholipase, it will decrease the phospholipids with the proteases, disruption of membrane and cytoskeletal proteins, and both will lead into a membrane damage. Also, it will activate endonucleases with a nuclear damage and ATPase, and this will lead into a decrease in the ATP. So this is the effect of increased cytosolic or entrance or influx of calcium into the cell. The ultrastructural changes of reversible cell injury and irreversible cell injury is demonstrated in this table. The reversible cell injury, as we said, the cell, it shows mitochondrial swelling with decrease in mitochondrial function, cellular swelling due to the effect of decreased ATP on sodium pump with influx of sodium and water lead into a cellular swelling with hydropic degeneration, formation of cell surface blebs, as we said, due to the entrance or influx of water and sodium, clumping of nuclear chromatin due to decreased pH due to an increase in the lactic acid. This will lead into a clumping of nuclear chromatin, detachment of ribosomes with endoplasmic reticulum swelling. Whenever there is severe mitochondrial damage with extreme damage to plasma membrane with loss of cellular contents, this is the stage of irreversible cell injury. Whenever we remove the stimulus, the cell, it will not return back to normal. Also, there will be a lysosomal swelling. Rupture of lysosomes will lead into the liberation of RNases, DNases, proteases, glucosidases, and this will lead into extensive digestion of both cytoplasmic and nuclear component. This is, we call it autolysis. It is a sign of irreversible cell injury and death. Formation of myelin figure, the dead cells, finally, it becomes replaced by large masses composed of phospholipids. Also, there will be a lysis of endoplasmic reticulum, while endoplasmic reticulum swelling is seen in reversible cell injury. And there is an important nuclear changes in irreversible cell injury. Pycnosis, condensation of chromatin, karyosis, fragmentation of the chromatin, and lastly, karyolysis or dissolution and lysis of the chromatin. This figure illustrates the difference between normal cells, the cells with reversible cell injury, and the cells with irreversible cell injury. This is this normal cell. This is a cell with reversible cell injury shows swelling of endoplasmic reticulum, mitochondrial swelling, clumping of nuclear chromatin with bleb formation. When we remove the stimulus or the cause, the cell will return back to normal. The cell, in case of irreversible cell injury, it shows bleb formation, formation of myelin figures, membrane blebs with rupture of lysosomes. Here, the lysosomes are intact. And also, there is a nuclear condensation or pycnosis with swollen mitochondria. And even when at this stage, even when we remove the stimulus, the cell, it will not go back to normal. The cell, it will show a cell death. Now it comes into irreversible cell injury or cell death. When we said irreversible cell injury, we will speak about irreversible cell injury in living tissue. We have two types. We have necrosis and apoptosis. I will speak about in the next lecture. What is necrosis? It is a sequence of morphological changes that follow cell death in living tissue. It affects groups of cells and always is associated with inflammation. The dead cellular contents elect a local host reaction called inflammation in an attempt to eliminate the dead cells. What are the morphological features of necrosis? Cytoplasmic changes includes the cytoplasm becomes homogeneous, intensely eosinophilic, sometimes it shows vacuolization or calcification. The nuclear changes, pycnosis, nuclear shrinkage, and increased basophilia with condensation of chromatin, karyosis with fragmentation, and later on dissolution or lysis of the chromatin. We call it a karyolysis. This figure shows the nuclear features of necrotic cells. This is a normal hematoxylin and iosine stain. This is a normal cell in which there is a nucleus. It shows an open nucleus with a nucleolus and pale pink cytoplasm. The early necrotic cell shows shrinkage and increased cytoplasmic eosinophilia. The nucleus becomes small and intensely basophilic, termed pycnosis. If the process of necrosis continues, there will be a fragmentation of the nucleus into several pieces. We call it karyosis. And the cell will show irregularity, and this irregularity in contour reflecting a cell membrane breakdown. The cytoplasm becomes more eosinophilic. The last stage of necrotic cells, there will be a dissolution or lysis of the chromatin, with the cytoplasm becomes deeply eosinophilic in color in comparison to the first normal cells. What are the fates of necrosis? The necrotic cells may persist for some time or may be digested by enzymes and disappear. The dead cells may be replaced by myelin figures, which are either phagocytosed by other cells or further degraded into fatty acids, which bind calcium and ultimately calcified. Now we will come into patterns of necrosis or types of necrosis. First type of necrosis, we call it a coagulative necrosis. It occurs due to sudden cut of a blood supply. The tissue retains the same architecture with loss of both nuclear and cytoplasmic details. Coagulative necrosis is characteristic of infarcts in all solid organs except brain. What are the pathological features of necrosis? Pathological features, we have to explain the gross features and histological features. The gross features of coagulative necrosis, the organ becomes pale, firm, swollen. Later it became soft, yellow, shrinked, boiled meat-like. Histological examination shows loss of a nucleus, eosinophilic cytoplasm, while the architecture of the organ retained the same. The best example is myocardial infarction. This is the normal myocardial muscles, this is the nucleus, and this is the fiber. And here this is a myocardial infarction or coagulative necrosis in which there is irreversible cell injury to the myocardial muscles. The myocytoplasm become more eosinophilic, loss of nuclei with presence of inflammatory cells. The architecture of the muscles retain the same. This is the features of irreversible cell injury in the myocardium or coagulative necrosis of the myocardium, and this is the gross appearance of a coagulative necrosis in the cortex of the kidney. The second type, we call it liquefactive necrosis, is caused by focal bacterial or fungal infection or hypoxic death of cells within central nervous system. The tissue becomes liquid, viscous, mass-like. If the process is initiated by acute inflammation, the material becomes creamy, yellow, pus-like, or we call it pus. Yellow, creamy, pus, we call it pus, if the primary cause is an infection. Tissue details and architecture are lost. This is the gross appearance of liquefactive necrosis, and this is an infarct in the brain showing dissolution of the tissue. The third example or pattern of necrosis, we call it caseous necrosis. It is most often seen in tuberculous infection. Grossly, the lesion is caseous cheese-like, which is a friable yellow to white in appearance. This is a hilar lymph node of the lung showing the caseation. And histologically, caseation looks amorphous, granular, and eosinophilic material. Fourth type of necrosis, we call it fat necrosis. We have two types, the traumatic fat necrosis, best example of trauma to the female breast. The second type, we have enzymatic fat necrosis due to release of enzyme lipase due to following pancreatic surgery or acute pancreatitis. And this is a fat necrosis. In the mesentery, it shows saponification. Fat necrosis with calcium soap formation or saponification at sites of lipid breakdown in the mesentery. This is the saponification, these whitish areas due to a fat necrosis. Lastly, we have fibrinoid necrosis. It is seen in small blood vessel and glomeruli in autoimmune diseases and malignant hypertension. Microscopically, there is a deposition of fibrin, which is a brightly eosinophilic hyaline-like deposition. This is the glomeruli, and this is the fibrin, which is brightly eosinophilic hyaline-like deposition. Thank you.